However, is that adult cells generally do reach a point at which their differentiation is complete, when they can no longer become any other type of cell. Stem cells and development. What are stem cells? One of the most important questions in biology is how all of them specialized differentiated cell types in the body are formed from just a single cell. Biologists say that such a cell is totipotent, literally able to do anything, to develop into any type of cell in the body, including the cells that make up the extraembryonic membranes and a placenta. Only the fertilized egg and the cells produced by the first few cell divisions of embryonic development are truly totipotent. If there is a secret by which cells start the process of differentiation, these are the cells that know the secret. Human development. After about 4 days of development, a human embryo forms into a blastocyst, a hollow ball of cells with a cluster of cells inside known as the inner cell mass. Even at this early stage, the cells of blastocysts have begun to specialize. The outer cells form tissues that attach the embryo to its mother, while the inner cell mass becomes the embryo itself. The cells of the inner cell mass are said to be pluripotent. Cells that are pluripotent can develop into most, but not all, the body's cell types. They cannot form tissue surrounding the embryo. Stem cells. The unspecialized cells form which differentiated cells develop are known as stem cells. As the name implies, stem cells sit at the base of the branching stem of development from which different cell types form. Because of their potential and develop into other cell types, stem cells are the subject of intense interest by researchers around the world. Embryonic stem cells. As you've seen, the pluripotent stem cells of the inner cell mass eventually produce all of the cells in the body. Embryonic stem cells are pluripotent cells found in the early embryo. In 1998, researchers at the University of Wisconsin found a way to grow these embryonic stem cells in culture. Their experiments confirmed that such cells did indeed have the capacity to produce just about any cell type in the human body. In fact, scientists have managed to coax mouse embryonic stem cells to differentiate into nerve cells, muscle cells, and even into sperm and egg cells. Recently, sperm made from embryonic stem cells were used to generate live mice. Adult stem cells. For years, biologists have suspected that an adult organism might also contain some types of stem cells. Cells in the blood and skin, for example, have a limited lifespan that must be constantly replaced. This suggests that the body contains pools of stem cells from which new skin and blood cells can be produced. Adult stem cells are groups of cells that differentiate to renew and replace cells in the adult body. Because of their more limited potential, adult stem cells are referred as multipotent, meaning that they can develop into many types of differentiated cells. Typically, stem cells of a given organ or tissue produce only the types of cells that are unique to that tissue. For example, adult stem cells in the bone marrow can develop into several types of blood cells, while stem cells in the brain can produce neurons or nerve cells. Frontiers in stem cell research. What are some possible benefits and issues associated with stem cell research? Understanding how stem cells retain the capacity to differentiate into so many cell types is an important unsolved problem in biology. Scientists would like to learn exactly which signals tell the cell to become specialized and how other cells remain multipotent. Potential benefits. Basic research on stem cells takes on a special urgency in light of the importance it might have for human health. There are many causes of damage to particular types of cells. Heart attacks destroy cells in the heart muscle. Strokes injure brain cells, and spinal cord injuries cause paralysis by breaking connections between nerve cells. Given the suffering and death caused by one of these conditions, the prospect of using stem cells to repair such cellular damage have excited medical researchers. Many hope to see a day when the damage caused by severe heart attack can be reversed using stem cell therapy. Experiments using animals suggest several approaches show promise of success. One approach might be to inject stem cells from the patient's bone marrow into the heart's damaged area, as shown in Figure 10.21. Another approach is to inject embryonic stem cells that might eventually differentiate into new heart muscle cells. Stem cells offer the potential benefit of using undifferentiated cells to repair or replace badly damaged cells and tissues. Ethical issues. Because adult stem cells can be obtained directly from the body of a willing donor, research with these cells has raised a few ethical questions to date. This is not the case with embryonic stem cells, which are generally obtained from very early embryos. Most techniques for harvesting embryonic stem cells cause the destruction of an embryo. For this reason, individuals who regard the embryo as entitled to the rights and protections of any human being object to such work. This concern has made government funding of embryonic stem cell research an important political issue. Groups seeking to protect embryos oppose such research as unethical. Other groups support each research as essential for saving human lives and argue that it would be unethical to restrict research. Humans' embryonic stem cell research is controversial because the arguments for it and against it both involve ethical issues of life and death. It is possible, however, that in the not-too-distant future, both ethical concerns will be addressed with technological solutions. Some recent experiments have suggested that there may be ways to extract a small number of the stem cells from an early embryo without damaging the embryo itself. Other experiments have shown that it is possible to switch on a number of genes that reprogram adult cells to look and function like pluripotent embryonic stem cells. Such a technique would do away with the need to involve embryos at all. It also might make it possible to tailor specific therapies to the needs of each individual patient. Approaches like these, if successful, might allow potentially life-saving research to go forward while avoiding any destruction of the embryonic life. The end.